Hello everyone, my name is Vasco Elbrecht and I'm a PhD student at the Ruhr-University Bochum. I am part of the GeneStream project, and in last May we traveled down to the Breitenbach. This is a famous small stream in Hessen in Germany, and there we conducted an ecological experiment. We used 64 of those channels to assess the impact of agricultural stressors on macroinvertebrate communities. In the next few minutes, I will walk you through the process of analyzing the data, and also we take a closer look at the samples which we took. Because of heavy rainfall and an upcoming flooding event, we started the final sampling of the channels at night. With the final sampling, we collect all the macroinvertebrates we find in the channels after a 5-week experimental period. We disconnect the channel from the water supply and transfer the complete contents of the channel into a tub. On top of the tub is a sieve with a 4 mm mesh size to retain all the big rocks. The channel is transferred to the sampling station. The container with the corresponding channel number is selected. It contains some ethanol to preserve the insects. The channel is rinsed with water, and the sand as well as small organisms are falling to the sieve into the tub. The gravel on top of the sieve is searched for bigger organisms like caddisflies or gammarids, which are transferred directly into the sampling container. After carefully picking out all the organisms from the gravel is discarded. The water and sand in the tub, which contains now all the small organisms, is poured onto a sieve with a 500 micrometer mesh size. This way we get rid of all the fine sand. But we keep the organisms. We are using hose to remove all the fine sediment. The coarse sand contains hundreds of small organisms which were in the channel. The sand is transferred with the organisms into the container using an ethanol bottle. By carefully shaking the container, we make sure that all organisms get conserved in ethanol. The container is returned into the box. Now you know how the final sampling of the channels was done. We are back in Bochum with our samples, and together with 3 bachelor students, it's now time to carefully analyze our samples in a very standardized way. In the next few minutes, I will show you how we analyzed our samples. We have stored our samples at -20 degrees, and all of them are in ethanol for later molecular analysis. Let's take a look at one of those samples. In here we have a lot of sand and plant material, and now the task is to sort all the organisms out of this channel here. We're going to go back to the lab and I'll show you how it's done. We have here our channel, and in each channel there was a leaf pack included, which is to measure the consumption rate of the organism. And this we put in a single tube. And then we have one tube where we put all the organisms we find in those channels. And we have one tube where we collect all the debris, which is plant matter and sand and things like that. So now I'm going to show you how we process those channels. We open it, and we're now taking a spoon, and we take some of this material here and put it on a petri dish. We are taking one of those grids as a black background. And now you can see already here some organisms which you can see with the naked eye. For example, over here we have a caddisfly, and we can now collect those with some tweezers. I'm now done sorting out all the organisms, which I can see by just looking at it, and I put it now under the stereo microscope. And I'm now looking through the microscope. In here we have those grid cells. I'm now going to sort this grid cell by grid cell. Now I move along each grid cell in a pattern like this. And this way I make sure I don't miss any organisms which are present in this sample. There are now no organisms left in this petri dish, so we take the sand and the plant debris and put it into the debris tube. When we have collected all the organisms out of our channels, the next step is to sort those on a family level. We're then going to use the statistical programming language R to analyze our data and to see the effects of the aggregate stressors on our communities. I would now like to thank you for watching, and we also thank our partners from the University of Otago who developed this experiment. And also we would like to thank the Bode Foundation for funding. If you want to follow up with this project and the results or the other projects that we are working on, you can visit our website genestream.de. Until next time, bye bye.